So good morning, everybody. Thank you very much for coming out to the 6th annual Waterloo Brain Day. And it's great to see people from across faculties and also from around southwestern Ontario. I know we have visitors at least as far away as Toronto. So it's good to know that people are coming to check out Brain Day in our 6th year. And I'm really excited that today we have world-class speakers once again who are all experts in their field, in fact, leaders in their field, and from across 4 different disciplines. And today, the order we will see them in is neuroscience, Gyorgy Buzsaki, theoretical neuroscience, Michael Hasselmo, psychology, Michael Tarr, and philosophy, Ian Gold. So let's just start by giving them a big warm welcome. Thanks for coming. So the topics that we're going to be exposed to today range from understanding some of the many oscillations found in the brain, to how face perception works, to how we come to think about the minds behind those faces. And on first glance, it may seem that the order of today's speakers kind of progresses from the neural to the psychological. But I actually think that most of the speakers would probably object to this characterization. And I think that all of them care really about integrating views across all of the different kinds of methods used to look at the brain. And if anything, that's really the theme of today's lectures, just like it's the theme of Brain Day, to try and get many perspectives on how neural function gives rise to the behaviors that we see. Before we begin the proceedings, I'm just going to sort of give you some administrative kinds of reminders. So there's going to be 2 talks this morning and then 2 talks in the afternoon with breaks, short breaks between each talks. There's refreshments that hopefully many of you have found already over in PAS 2085. For lunch, there's about an hour-and-a-half break. So for that, you're on your own. If you're not from around here, then probably following the majority of people somewhere is a good idea. There's lots of food on campus. Actually, I think the restaurant upstairs is closed. But there's also a university plaza kind of over that way, which has lots of restaurants which will be open. After the talks, there's a reception in the Center for Theoretical Neuroscience, which is in the new wing of the PASS building, 2464. So that will start at about 4:15, and everyone's invited. The first 30 or so people will get some delicious door prize, and everyone else will be near the refreshments, for which they can pay on their own. And actually, also, Paul Thagard reminded me yesterday that for the graduate students, there's a meeting of the Cognitive Science Grad Association afterwards in the green room in the grad house. And the speakers and other people will go have a nice dinner somewhere. The last thing, I will just point out that I'm going to be sending a sheet around. If you'd like, you can write your email address on there, and we'll add you to the mailing list for the seminar series for the CTN, so you'll get updates on speakers that we have coming on a regular basis. So finally, I'd like to express my thanks to the Faculty of Arts and the Cognitive Science Program and the Center for Theoretical Neuroscience who are funding the event. And perhaps most importantly, I'd like to thank Debbie Dietrich, who is the administrative assistant who's made sure I didn't mess anything up and made sure that all of our speakers got here on time and did all the work. So without further ado, let me proceed to the first speaker of the day. It's my very great pleasure to welcome Dr. Gyorgy Buzsaki. He is the Board of Governors Professor at the Center for Molecular and Behavioral Neuroscience at Rutgers University. His exceptional career over the last 3 decades, which included a postdoc just down the road at Western, has seen him publish over 200 scientific papers and books. Much of this work has been dedicated to understanding the hippocampus, though more recently he has been looking at interactions between hippocampus and other brain areas. A major theme of his work has been to understand the oscillatory features within and between brain structures. And his recent highly regarded book called Rhythms of the Brain has been praised as, quote, a must-read for anyone interested in understanding the functioning of large and complex circuits in the top journal Nature. We are extremely lucky today to have one of the world's foremost experts on neural oscillation speak to us. Please join me in welcoming Gyorgy Buzsaki.
Thank you, Chris. This is wonderful.
Despite the late flight, you're up early.
As Chris mentioned, I did a postdoctoral fellowship not so far from here at the University of Western Ontario. And that was the year 2000. It was one of the heaviest winters, and we had very, very large amount of snow. And I just remembered that we had about 4 or 5 days when we were in the lab, many of us, and couldn't venture out. Every single day we flipped a coin and decided who is going to get some food. And I also remember that after that summer, we went to Montreal with my wife and visited several places, and we drove through Waterloo. I'm almost confident, although not 100% sure. Anyway, the reason why I can recall all these interesting stories is because I have a mechanism in my brain. I've got a search engine in my brain that allows me to travel back and forth in time and recreate the experience, at least the spatio-temporal conditions of those experiences. In fact, the essence of my talk is the following. If nature invents an algorithm for something useful, such as navigating in real space, that algorithm can be used for something else in larger brain animals, such as us, for something else, such as navigating in cognitive space. And this is what we call recall or planning for the future. Now, before we get there, we have to say, why is the hippocampus a search engine? Why is it so effective? It has to have special structure, and it has to have a special function. So let's see what the specialty of the hippocampus is. If you look at this brain, and you can say that there are at least, or we can state, 3 types of organizations here. One, what you can call strictly local organization. Everything is next to each other. They are not talking to each other, just like you now sitting in this room. It doesn't require a lot of wiring. In fact, the cerebellum doesn't even have a corpus callosum. It doesn't have long-range connection. There's very little white matter there. So you can put in lots and lots of neurons, and they are running really parallel, and only local computation is going on all the time. Now, opposite to this is this random network of the hippocampus. You can object already that there is no such a thing as random network in the brain. But if you are looking for randomness, the CA3 region of the hippocampus is your best bet. Now, in between these two extremes is the modular structure of the neocortex. And I don't have to have a better word than use Steve Strogat's term, the small world network, where local and global are working together. That is, most of the modules are locally organized, most of the connections are done locally, but there are several and enough long-range connections to do the following thing. The essence of cortical computation is that whatever is computed locally is broadcasted globally. Vice versa, the local computation is always under the control of the global state of the brain. Now, let's see out of these only the hippocampus, and you will see that the hippocampus is not functioning alone. We divided the labor with Michael Hasselmo. Michael will talk about the equally important areas of the limbic system, the entorhinal cortex and its colleagues. I'm focusing on the hippocampus. Now, what you see here is a single CA3 pyramidal cell impaled in vivo, filled, and reconstructed in its completeness. It's a little sad in this area of connectomes that this is one of the handful fully reconstructed neurons in the mammalian brains. But if you look at this one, it's quite remarkable. It has about 0.5 meter of axons and has about 40,000 boutons, which gives you 40,000 potential contacts with other neurons. But of course, this neuron is not by itself. It has another 200,000 or so partners in the hippocampus. So if we put this one neuron into the right location, you can see that this one neuron can communicate with almost 1/2, sometimes 2/3 of the entire structure. The hippocampus seems to be a single giant cortical module. Unlike the modular system of the neocortex, this just can grow. And then, of course, there are differences between dorsal and ventral, but let's disregard it now. So when you have so many cells, then those have to be connected. In order to connect 200,000 cells, it would require no more than 16 connections to make what we call a complete graph. So then you can have every neuron connected to everybody else. And it would be like the map of Canada, that you can go from Waterloo to Vancouver, but you have to go through the Trans-Canada Highway, which I did, and you have to go step by step from one place to another. In other words, you have to go through several computational steps. Now, for Neumann suggested that those machines or those devices that require multiple computation steps, they have low logical depth. In contrast, those computational devices that require only one or two or few computational steps have very high logical depth. The hippocampus has a very high logical depth because instead of one neuron communicating with 16, one single neuron can communicate with 20,000 or so CA3 other pyramidal cells. It's an incredibly strongly connected graph. Of course, it costs a lot of wiring and a lot of energy to maintain this. You look at this wiring. This is 40 miles of axons in a hippocampus in the rat, which is only 10 millimeters long. So what does it buy you? What it buys you is that you can go from any synapse to any other synapse in just 2 steps. Or you can go from any neuron to any other neuron in 2 steps. So the logical depth is given. So this search engine is extremely efficient because you can visit all the items very effectively and works like a librarian that it's not enough to have all the information in the world in books or in your neocortex, but you need a librarian who will point you to the right shelf and perhaps the right book to find the right kind of information. Are you still with me? Okay. Do you have any objection? You should, because I said something foolish. What I said is that you can go from anywhere else, from one neuron to 20,000 neurons, and then from 20,000 neurons to another 20 times 20,000 neurons. Well, that doesn't work. The reason why that doesn't work is because these neurons are connected with each other typically through one single release site, one single synapse, and so these are AMPA receptors. And AMPA receptors are notoriously weak. So there's absolutely no way that a single pyramidal cell can discharge its 20,000 partners. So we need something else. What we need is that many of these neurons, or at least a group of these neurons, have to come together into a coalition, a temporal coalition, that with a Canadian word we call cell assembly. So neurons have to come together in cell assemblies. So we need to generate synchrony among them because that's what matters for the downstream neuron. So how do you generate synchrony in the brain? One easy way is this. This is not even a neuron assembly. This is a performance in Budapest, which was pretty good. And after the end of the performance, people clap their hands, and sometimes in an interesting way. So you could see that the clapping became rhythmic. This is not only a cultural phenomenon, but it's also a very interesting phenomenon for physicists to understand. How is it emerging? There is no conspiracy, there are no leaders, there is no, nobody who has 10 times larger hands than somebody else and would make a difference. It emerges. But when it emerges, it generates these interactions among the individuals, generates what it's Hermann Haken, the laser physicist, suggests this is an order parameter. And once you have an order parameter, then the members are enslaved. Their degree of freedom is decreased. Therefore, they have no choice. They have to clap their hands rhythmically. So what do you gain? What you gain is noise and the surge of noise. And of course, the entire goal of clapping your hands is to make noise. It is so efficient to clap your hands rhythmically because the surge of noise is very high. So this is a very good thing because it doesn't cost anything. The energy that allows you to synchronize with rhythms has no cost involved. And this is exactly what the brain, or the networks in the brain, exploit, of course. What we need is that at least a group of neurons come together in a short period of time, which I identify with the integration time of the postsynaptic pyramidal cell, which is about 10 to 30 milliseconds, which is about the GABA-A time constant, which is about the GABA-A receptor time constant, which is about the AMPA receptor time constant, which is about the time window of a spike time independent plasticity, and which is about a gamma cycle. So this is a fast oscillation. So it is not by chance that these neurons come together, because their entire goal is to discharge the postsynaptic neuron. So in order to prove what I just said, we need to record from large number of these cells. And I already can imagine there is a whole entire orchestra here in the brain, and we have to identify each of them as individuals. And instead of putting in wires, which I used to do in London, Ontario, now these days we put in silicon probes, so we can, with the silicon chips, we can record from large number of neurons simultaneously. So it is one recording. This is an example. So this is the output what you get. So the slow oscillations, the slow patterns that you can see, are the summed membrane potential changes of all the neurons nearby. And then you see these sharp deflections, which are the outputs of these neurons. These are the spikes. So from this picture, through various tricks, we identify these individuals, such as, this is the pianist, and this is the drummer, and so on. And then once you are done with identifying the individual players, then we group them at least into two major groups: excitatory neurons and inhibitory neurons. Okay? And then, of course, when you have so many neurons, you've got a lot of problems, the same kind of problems that happens with clapping your hands and understanding how the interactions occur. But let's start with a single example. The single example is the following. This is a rat carrying a little LED on the head, and the animal is exploring the environment. And you can hear that there are spikes at this point. The spikes can be converted into color, and you have a tuning map of this neuron. This is a hippocampal CA1 neuron. This is a fantastic discovery, and this one single movie or slide is trying to summarize 30 years of very, very hard work of the very best people in system neuroscience. But if you listen to this neuron carefully, you can hear that it fires rhythmically. In this particular trial, it fired at 8.7 hertz. So now we have an interesting thing, namely that we have a single cell oscillator, which oscillates at this particular frequency, and we have many neurons that contribute to the field, whose membrane potentials contribute to the field, and they generate a rhythm, let's say 8 hertz. So we have two oscillators with non-integer relationship to each other. If you generate such a pattern, or if you have such a relationship, you generate interference. Now, interference equals precision, timing precision. So out of this very simple observations that took 30 years, two major things came out. In fact, one. Namely, John O'Keefe suggested that the hippocampus, because there are many of these cells there, hippocampal pyramidal cells are place cells. They fire at a particular part of the environment. Many neurons together, they fire in different parts of the world, so they generate a spatial map. And this spatial map allows the animal to navigate in the environment. And in order to identify where you are, the neuron has to fire, and then you can integrate the activity. So the way, or the reason why this neuron fires, according to the initial thoughts, is that the environmental cues, the constellation of the various modalities, make the neuron fire. So for example, when the animal is going, well, passing by this corner, then the attributes of this corner somehow, by some means, activating the entorhinal cells, grid cells, and other neurons, eventually converge onto the hippocampus. And this is the cause of the firing of this neuron. And when the animal passes the corner, then the firing rate goes down. Of course, it takes some time to integrate over this period, so it would take about a half a second to a second. So this is not a very precise code, it's a rate code. But O'Keefe also discovered something interesting, which remains probably the most powerful argument for phase coding or temporal coding in the brain. Namely, that you don't have to look at this large period. All you have to do is just take a snapshot of a theta cycle and look at the relationship between the phase of the the spike and the theta wave. What you can see is that this neuron fires at the peak of the theta cycle. When the animal enters the field, it fires at the trough when the animal is in the middle, and then it will continue to precess when the animal is leaving the field. When this was discovered, I became super enthusiastic about this because time entered into this very interesting place field. To my astonishment and to some disappointment, O'Keefe argued that this is one more argument in favor of the spatial mapping theory of the hippocampus. Namely, that this phase tells the animal where the animal is, therefore it is a further proof for place code. Of course, if this would be the end of the story, then I wouldn't be able to figure out how I remembered my days at University of Western Ontario. Because there are no external cues when I do the remembering. I can remember things without any external guidance. And the way it works is very simple, said Donald Hebb. Namely, something happens, an assembly of neurons come together, they live for a while in the dorsal hippocampus, typically for a second, they die or they dissipate, and then another assembly is activated, which activates another set of assemblies and another set of assemblies, and this chain of assemblies or assembly sequence can go on forever. I'm telling you, I'm giving you this talk, even if I don't have slides or anything, I'm not even looking at you, I can give you pretty much the same trajectory, because the information in my CA3 n-dimensional system can go in always in the same trajectory, same direction, given the same initial conditions. Okay, so that's the fact. This is the summary, and I told you already the problem. And the problem could be formulated this way also, is that when you have multiple of these neurons, and they are sequentially active when the animal travels in the environment, what is the cause of this sequentiality? The simplest explanation, of course, is that the outside the hippocampus, outside the brain, the environment is causing the neurons to fire in a sequence, one after the other. But there is another way of thinking about this, namely, no. In the case of memory or recall or planning into the future, you don't need that. Instead, what you need is an internally generated sequence generator or sequence events. So let's test this two hypotheses. And the good thing about, of course, good theories, such as O'Keefe's theory, is that they are explicit. The better, the more explicit the theory is, the better the theory is. So there are a couple of conjectures. Namely, if in theory you could freeze an animal here and now and maintain the dynamic of the theta cycling in the hippocampus, then a subset of CA1 neurons, CA3 neurons, entorhinal grid neurons, and all those neurons that dedicate or define the here and now should fire forever, and everybody else should be shut off and completely silent. Those few neurons, they happen to be activated continuously, should fire always on the same phase, according to the theory. And of course, this is not happens. That's why I'm here. So let's just do something easy first. Namely, what we do is fix the head of the animal. In this case, it's a mouse. So you can see that this is a belt that is not driven by anybody else except the mouse. It's the only thing that moves around actively, or moves around, is what is being actively moved by the animal. The environmental cues are completely inert, but we made the local cues rich and available. So you can see a little forest here, then there are little plexiglass cubes and things like that. And one of them predicts that there will be water. So the animal runs 1.6 meters or so, and then there will be a water reward, and it starts all over again, all over again, all over again, as if the animal would be running this alley and then would be teleported back, and so on and so on and so on. So, of course, the important thing to ask is what happens to the hippocampal cells. Remember that according to the O'Keeffe's theory, only a subset of neurons should be active. Everybody else should be silent, but the subset of neurons should be active throughout the trials. There is no one neuron that satisfies that condition. Instead, this is the belt, 150 centimeters, and these are the individual neurons. They were recorded simultaneously. There are 75 of place cells, if you want. And you can see that the entire journey is represented, and the lifetime of these neurons is pretty much the same from the beginning to the end. Now, this is just one trial. This is the next trial. I have many trials. And then when you look at the session average, then you can see that, indeed, on average, the lifetime, that is how long the neuron and its partners that we are not recording last, is about a second or so. So you can say, it's not the distal cues, it's not the landmarks that matters, but probably local cues. So now let's try to eliminate those. And then we go to the rat. In this situation with Eva Pastalkova, what we did is a simplified version of this. Now we involve the hippocampus also, because the task is to go to the right, get a water reward, and when the right side was rewarded, you have to go to the left. It's a spontaneous alternation task. In between the trials, the animal has to wait for about 10-15 seconds, but we wanted to make sure that the animal is not chewing the cable or doing something totally useless activity. We asked the animal to run in a wheel, always facing in the same direction at the same speed. In fact, the very first running wheel experiments I did at the University of Western Ontario in 1980. So this came back to me and said, and asked several colleagues, what is your prediction? What will happen if the animal is running now in the wheel? Now, we have done everything in our power to reduce and make the environmental cues constant. The local cues are constant, as well as the idiothetic information from the body is also constant. Everything is constant. So let's see what happens in the hippocampus. Again, theory suggests that there should be neurons that fire continuously throughout the trial and in the same phase. Instead, what you see here is that now it's not distance, this is time. And there is a neuron, this neuron here, that fires at about 2 seconds and every single trial. There is this other neuron that fires at about 5 seconds at every single trial. And there are many of these, many patterns like this. You can collapse this firing activity into a session average. And then this is 58 neurons recorded simultaneously. And you can see that, there is an entire sequence, just like in the mouse. And if you have enough neurons, every part of the journey is represented perfectly, even though the entire thing is smooth. There are no landmarks, no specific things here that would be used as a cue. It seems that everything is just generated equally. Moreover, if I record and look at the lifetime of these, that each of the neurons of these neurons are active for about the same amount of time. So if I give this spike train to, let's say, Michael, and said, what happened to this animal? He or anybody in the field would say, I think the animal is navigating in the environment, because the pattern is exactly the same, except the animal is not going anywhere, physically. So the next step, of course, if you look at any of these active neurons, then you will see that every neuron that is reasonably active, meaning at about the theta frequency, which is about 8 hertz, undergoes a phase precession. So this is, again, time. This is the activity of the neuron, and this is the phase of the theta cycle. And you can see these nice diagonals here. Again, violating the rules. Now, the good news about this task is that the information about where the animal was coming from and where it is heading to, that is the past and the future, must be in the hippocampus. We know it because other people have done lesion experiments, and without the hippocampus, the animal cannot perform this task. So it means that there should be a rule, there should be a sequence pattern, there should be a trajectory of activity of this sequence activation that is unique and specific to a particular direction. So instead of showing you many neurons, let me show you one at a time. So this is just one neuron. Oops. Again, there is an LED on the head of the animal. This there's a spiking activity distorted by the O amplifier. The animal went to the left. So the neuron fired, and the animal went to the left. Now it's not firing. In fact, it's completely silent, and the animal went to the right. It's firing again. So we already have an observation. We have to verify it. We verified it. Yes, if it's firing, it goes to the left. If it doesn't fire, then you can already tell me what happens. Which direction? Come on, it's still early morning. The animal went to the right. Now the animal came back, it's firing. Which direction will it go? Left. Okay. All right, so we now have a routine, right? So the animal goes back, there's no firing. And even those of you who are still dowsing, they know that next time it should go to the right according to our prediction, according to the history of the behavior, right? But it's not always so simple, because this animal is running longer than 15 seconds now, and something is unusual happening here, such as these two spikes, and then another two spikes. So the neuron is spiking, and if you believe the neuron spiking, you will say the animal should go to the left. So the question is whether we follow the behavioral prediction or the neuron, and we follow the neuron's prediction. And we go back. Of course, this is very nice. It was running continuously. We know that this is a trial which is predicting the animal has to go right, because it was a correct trial. The previous one was an error. The animal goes back to the wheel. It's firing. Which direction does it have to go? It has to go to the left. No, it goes to the right. It comes back. The neuron fires, reminds the animal that you have to go to the left, and the animal goes to the left. So this is all fun, and this is our neuron, but it's absolutely useless to guide behavior. The reason why it is useless to guide behavior, of course, is because this neuron lived only for a second. And there is another 19 seconds that has to be gapped before the animal actually comes to what you call a choice point. So it has to have partners, and it does have partners, such as this neuron, this neuron, this neuron, this neuron. So there are multiple sequences. What we think happens, of course, is that this neuron gives this information content to this neuron, and to another neuron, and another neuron, and another neuron, and it can go ad infinitum. In fact, O'Keefe suggested that if you let the animal go from Waterloo to Vancouver, then on the journey, every single one of these 250,000 CA1 pyramidal cells should be active somewhere. That is, all neurons are place neurons. They should be active at one time somewhere in some constellation of the environment. We can claim the same thing. If we don't let the animal travel, but the animal would be running 24 hours continuously in this wheel, then every one of the neurons in the hippocampus, in principle, should be active at one point somewhere. So if we have many of these neurons, then the thing that we can do, just like with the single neurons, say, let's select the same neurons that we record from according to the trial outcome, whether the animal in the future will go to the right or it will go to the left. So these are the identity of the neurons, which are the same here than here, except here the animal made a left choice, and here the animal made a right turn. And you can see that you can slice the time in very narrow windows, and it doesn't matter where you do it, you will always, if you have enough neurons, you will always get an intelligent prediction which direction the animal will go 20 seconds down the road. So we can predict the behavior of the animal 20 seconds ahead with 90% accuracy, including errors. So we can tell with high accuracy that the animal will make an error just by looking at the activity of the hippocampal neurons. Now, once you are done with this, then we come to a point that we love to do in the lab, which is rejecting ideas. So at least we can reject one exclusive explanation, namely that, or put it this way, I'd like to give credit to all those people who have done very beautiful work in this field, that their work is not discredited, their work is amplified, in fact, by these findings. Namely, it suggests that the hippocampus can generate sequences in two different ways. One is that if there is no input to the hippocampus, but there is the dynamic, the theta dynamic, it just can't help. It will generate sequences forever. On the other hand, if there are salient or whatever those words, appropriate words are, or important cues outside there, then it can change the trajectory in any direction. And that's what happens when the animal is navigating in the environment, and there are environmental cues that allow an update. For example, if I would be telling you a story and Chris would interrupt me with questions all the time, then my trajectory would jump. And it would be eventually his story and not my story, because the outcome would be very similar to his wish rather than to my goals. And this is the nice thing, that in the hippocampus, this trajectory can switch in just one single theta cycle. In a 100 millisecond time frame, you can jump and update the information with the help of the internal codecs very effectively. Now, because this is also a computational course or computational day, let's see something more exciting. Namely, that the animal speed, of course, is not constant. So let's just consider two trials when the animal is running slow and twice as fast. And this is the place field size of the neuron. Here, just the size. And you can see that during slow run, it takes 10 theta cycles to finish to go through the place field. In the second trial, it takes only 5. But if you count the number of action potentials, they are the same. In fact, if you count the action potential in every single trial, slow or fast, it doesn't matter, then they are pretty much the same. So the resources, the number of action potentials that are dedicated to code the environment, if you want to code anything, can be played or replayed in a few cycles or multiple cycles. Now, this is very interesting, of course, because if you look at the frequency of the oscillation, that is not the frequency of the firing, but the frequency of the packages of these spikes, then you can see that if the animal is running fast, then the oscillation frequency of this neuron is faster. Every single neuron in the hippocampus and in the entorhinal cortex are speed-dependent oscillations. Or, if you want, attention-related oscillation. Attention can do exactly the same thing. Now, at the same time, as I learned it from Kees van der Wolf when I was in Canada, the theta frequency changes a little bit, but just a little bit. So it's almost negligible in this comparison. The result of which is very cute. Namely, we have an oscillation frequency. That is, we have time. We have speed, or some neurons, or some computational circuit that Michael will talk about in this hippocampal system has access to the speed signal. Then we can compute distance. So the reason why there is a nice relationship between the phase of the theta cycle and where the animal is not because the environmental cues determine the phase, but it's because of this temporal code that allows distance translation. Now, one step further, if you look at many neurons, in this case about 1,000 CA1 neurons, or place cells that are active, every one of them oscillates faster than the ongoing theta rhythm. And now we have a big problem. The problem is that theta rhythm is generated by these neurons. Their membrane potential fluctuations are the ones that generated extracellular currents. So how is it possible that everybody oscillates, let's say, at 9 hertz, and everybody's clapping their hands at one particular frequency, but in a tricky way, namely that for a short period of time and with an increasing amplitude, decreasing amplitude. But what I would hear is slower, let's say 8 Hertz. And we cannot blame it on the input of CA3 or the dentate gyrus or the entorhinal codex layer 2, because they do exactly the same. Every grid cell and every place cell in the hippocampus oscillates faster than the ongoing theta cycle. So we have to solve this somehow. And the solution is extremely simple. An assumption. So, of course, we have to prove the assumption. But if you understand the following assumption, everything falls into place. Namely, all you have to assume that there is an order between how you clap your hands and your neighbor. That is, time is running on multiple scales, in this case at least on 2 scales. One is what you can call real time, that the real time is how long time does it take for the edible to go from place A to place B, the representations of these 2 cells. At the same time, because these neurons have long and heavy tails, they fire together almost anywhere, including every single theta cycle, if they overlap. And there is a temporal relationship between the within theta cycle, in the tens of millisecond scale, and which we call tau, and the distance or the temporal distance it takes for the animal from going from here to here. In other words, all we need is that there is a linear relationship between long time scale and short time scale. So, for example, we have 3 oscillators, not even neurons, and we can see that the T here has a correlation with tau. And if we have this rule, and we have about 200 neurons or 200 oscillators together, every one of them oscillates at 9 Hertz. The envelope, that is what you would integrate with your ear, if it would be clapping, would be 8 Hertz. It's almost magic, right? The magic, of course, is, and I can change this frequency and slow down to almost any number if we introduce or make the tau longer and longer. That is, the delay within the theta cycles longer and longer. What comes out of this, inevitably, is phase precession. In other words, every neuron is oscillating relative to the conglomerate product of everybody else. And there are lots of interesting things that come out of this. Namely, if you stick out your head from the crowd, then the crowd pulls you back, typically, because they want to equalize. Same thing happens with the place cells. If the cell oscillates very fast, its lifetime is very short. The place field is very small. The phase precession slope is small. To make the long story short, it's published data. There are multiple things in the place field literature that characterize what's happening. For example, the place field size, the peak of the place field, the slope of the phase precession, the oscillating frequency of the neuron, and so on and so on and so on. Give me 2 of those, I can give you all the others in any combination. So this rule must be persuasive. This rule must be very important. Is this true? That's the question. So let's go back to the real world. Now we are recording from 2 neurons simultaneously. And this is our rat. And the length of the blue thing shows how fast the animal runs. Sometimes it's fast, sometimes it's slow. And this is the distance representation between the 2 neurons. And this is the temporal offset between the green neuron and the red neuron. And you can see that there is an 80 millisecond or so time lag here. And the peak is always at the same time, irrespective of the animal's speed. So the speed of the animal changes, but the temporal offset between these 2 neurons does not. Now let's see more neurons. So this is we have 5 neurons. The animal is running in this direction. Every dot is a neuron, a spike. You add them up, and you get these nice tuning curves. 1, 2, 3, 4, 5. Now you look at the temporal relationship of these neurons in a single theta cycle, and you will find that, I look at neuron 1. This is my reference neuron. Neuron 2 is second in the real time, but it's also second in the theta time. in the 10 millisecond or tens of millisecond timescale or so. So what you can do now is measure the distances in the physical world out there with a ruler, measure the temporal offsets in the temporal dynamics during theta activity, and cross-correlate them. And you got a very nice linear line here. What does it mean? It means that, with some simplification, in the dorsal hippocampus, let's say 10 centimeter distances between two neural representations means that they are 10 milliseconds from each other in the theta cycle. And A is before B. If it's 30 centimeters, it will be 30 milliseconds. If it is 50 milliseconds, then it corresponds to 50 centimeters. If it's 100 centimeter representation distance, then it's 100 milliseconds. And this is very good, except when you have many, here we had only a handful of cross-correlograms. Now, if you have 17,000 cross-correlograms, then the picture changes, and the linear becomes sigmoid. So why does it become sigmoid? So I said 100 centimeter corresponds to 100 millisecond. How about 150 centimeters? It cannot correspond to 150 because it's longer than the theta cycle. So those things that happen to be longer or further apart in time and in distance cannot be part of the same story. It cannot be integrated by a single theta cycle. So this is the reason why there is a sigmoid here, because this is the duration of the theta cycle. Which means that this part of the environment, let's say it's a meter or so, is beautifully resolved by this dynamic in a single theta cycle. But everything between, let's say, a meter and a half and infinite is lumped together. In other words, what may have happened, this is sort of good news also for psychology, is because what usually happens that information from my long-term memory comes up and goes into short-term buffer, and this is where the clarity of information is enhanced. So for example, I remember the previous slide and probably the next two slides that are coming up, but I have no clue what my first slide was, or just vaguely, and the relationship between those slides and what's coming in the future. And the time can always zoom in, and a very large amount of information can always be compressed into these single theta cycles. So for example, I can ask you about a book, and you say, oh, the book is about this in 5 sentences. Or I ask you, what is chapter 11, or chapter 10 is about? And then you can say another 5 sentences. What's on the second paragraph? And if you remember the book, so in other words, there is no real definition of episodic memory. Any of the above is episodic information, because a sentence could be an episode, and your entire life story could be an episode. There is no beginning and no end, except that the resolution, the clarity, is always defined by some temporal rules, such as the one I'm trying to explain you. So speed didn't matter, remember. Now let's say we can do another trick. Bruce McNaughton and also Neil Burgess and his group have already shown, both in modeling studies as well as in real situations, that if you put the animal into a larger size environment, the place fields will move apart a little bit. So now we can do a trick and ask what happens if the animal is running in a 1 meter track, and then all of a sudden you double the size. Then we know what happens from these previous colleagues is that the depth representation of neuron A and neuron B will be longer. So instead of 50 centimeters, it will be 70 centimeters. The critical question is, of course, what happens to the temporal offset of these neurons? Will they follow? So 50 milliseconds becomes 70 milliseconds or not? And the answer is no. Irrespective of the size of the environment, the temporal representation, or not the representation, the temporal relationships of these neurons remains pretty much the same. Now, this is a profound claim, because it means that the fundamental organization of the brain, or perhaps just at least now for the hippocampus, is temporal. Everything else follows from it. Namely, if you put the animal in a small box, then these same resources allow very high resolution of the environment. In other words, the place fields will be very small. But if you put the animal into a football-sized field, all of a sudden, the resolution of the environment will suffer. The same exact way how I just explained when you have to summarize a book versus when you have to summarize a single paragraph or a single page. So to put things together, this is the sort of a summary that of what I had said so far, is that the animal is running through the place fields. It takes about a second or so in the dorsal hippocampus to finish or go through the place field. And that takes about 7 theta cycles or so. So every neuron is active for about 7 theta cycles if everybody else is equal. Which also means that in every single theta cycle there are about 7 plus minus 2 possibilities, as John Lisman exploited this further, that indeed the here and now is always represented by the most active cell assemblies, which is in the trough, with the help of other assemblies which were most active before or which will be active in the future, and they are on the descending phase or on the ascending phase. In other words, every time slot of the hippocampal theta cycle is exploited, and the quick question that comes from this is whether this is useful for something. Did anybody understand what I was trying to say? Not one. There is one person who is disqualified. Okay, well, at least besides Michael, there was somebody else who did. And this was Sebastian Bach. Just let's assume that this is a place cell. You know, the firing rate, it just goes up. And of course, this is a fugue. And Bach fugues are absolutely marvelous. You know, it's unlimited what they can perform. So this is our place cell 2. We don't have time to go through the entire fugue. It's a beautiful piece. But so then the third place field comes in. And then you will see something remarkable, namely that there is a nice relationship between the long temporal play. This was the first one, second one is that you see a very nice short-term relationship. The essence of music is that it runs on multiple timescales. That you sort of can predict what comes next from these short-time temporal relationships. And this is probably what Bach and many other musicians exploited, because this is pleasing for the ear. The reason why it is pleasing for the ear is because this is the way how the brain is organized. At least this is the way how the hippocampus is organized. So now if you believe what I had just so far, then you would like to understand the most important question of this question bank is that what keeps these neurons apart in the hippocampal theta cycle? Why don't the assemblies just fire together? Neurons can fire together, such as they do in epilepsy. So what keeps them apart? I give you the clue, or the hypothesis perfectly. It's perisomatic inhibition that allows that neuron sequences or assembly sequences are spread apart and they don't fire completely together. So let's prove it. And I think I have to rush a little bit. I skip through this. Now we have to go back to the mouse. We have to use contemporary methods, such as optogenetics, because what we would like to do is silence the perisomatic parabobin-containing interneurons. And as a contrast, we are using another construct, which is the somatostatin neuron, that targets the distal dendrites of the same kind of pyramidal cells. So in one case, in one experiment, we can shut off or remove perisomatic inhibition from a small group of neurons in the hippocampus, leaving every part of the circuitry intact. In another group of animals, the perisomatic inhibition is intact, and we remove the dendritic targeting inhibition. And this can be done with yellow-green light and with halodopsin that is in these specific neurons. And all we have to do is we have to edge the silicon probes or the lights and put on the silicon probes and place it in the hippocampus and do the same kind of experiment that I had shown you previously in the running belt experiment. When you do that, you will find the following. These neurons are silent when you shine light. So the way it works is that this is 150 centimeters, this entire belt. In the 1/3 of it, and from one object to the next object, the light is on at every second trial. So one trial is nothing, second trial is inactivation. Nothing, inactivation, nothing, inactivation. And this is what you see, the contrast here. And these are the place cells or pyramidal cells, and we find that inhibition inhibits. That's not exactly a big discovery, but it does it in a very interesting way. Namely, now we separate the place cells in such a way that these are the ones that are active in the light area, and these are the ones that are active outside the light area. That is, they hardly ever fire a spike when the light is shining. And you don't see a difference between the control and the light trials, which means that taking away perisomatic inhibition does not make neurons fire. So the reason why neurons don't fire in vivo is not necessarily because their soma is actively inhibited all the time. And one potential explanation is that they are sitting very close, their membrane potential, the resting membrane potential, is very close to the chloride equilibrium potential. So whether there is GABA inhibition or not, it doesn't make much of a difference. However, when the animal goes into the field and the cell becomes active, this is when inhibition becomes very important, because here you can see that the spikes are more when there is no perisomatic inhibition. But that's also not maintained steadily. In fact, how much time do I have? I have to rush, right?
You have till about quarter after, actually. And then
Okay. So what you are asking is that, is this inhibition just continuous, constant, or it is changing? And the answer is it's changing dynamically in the following way. When the animal enters the field, perisomatic inhibition is strong, and it's getting weaker as the animal goes through the place field, as shown by the black line here. What we have done here is that this is a normalized place field, all the place field we have, and divided them into 5 bins. And you can see that bin by bin, the effectiveness or the power or the control, the inhibitory control of the power movement cells go down. At the same time, the strength of the dendritic targeting inhibition is increasing. So this was weaker, we're getting weaker, this is getting stronger. There's a very nice division of labor between these two types of neurons. And needless to say, this is only 2 types. Peter Somogyi describes about 22 different types of neurons whose main job is nothing else, just control timing, or do something with the inputs, or control the outputs in a very complicated and not very well understood way. So what do the somatostatin neurons do? Well, they do something very important. If I ask many of you who are working on slices or some other preparation, what would happen with the firing patterns of the neurons if you disinhibit them? Then you would say, oh, the neurons would burst. Typically, that's what happens when you flood the slice with bicocooline, because the entire system is active. It's like, the hand clapping will emerge. On the other hand, if you just do it for one individual, you deafen only one individual, it doesn't have an impact from the others, then it turns out that perisomatic inhibition has very little impact on the burst firing pattern of the neurons. However, if you remove dendritic targeting inhibition, then the neurons start bursting like nuts. They really become true and very powerful bursters. In fact, the reason why after somatostatin disinhibition, the firing rate goes up is not because there are extra spikes here and there, especially not outside the field, but the neuron bursts a lot more. But the most critical thing, of course, is this. Now, Mice are a little different when it comes to phase precession. And remember that we have to look at only the first 2/3 of the place field, because here power-over inhibition is already very weak. But the phase precession is very sharp and large in the intact situation. When we apply parvalbumin light and decrease perisomatic parvalbumin inhibition, then what you can see is that the spikes at the beginning of the field, which should fire at the peak of the theta, they move to the trough. And when the animal passes by and they should fire on the rising phase, instead of rising phase, they move back to the trough. In other words, in the absence of perisomatic inhibition, the spikes tend to move to the trough. Synchrony is becoming stronger. In the other group, in the somatostatic group, whether there is dendritic inhibition or not, it doesn't matter. The timing of the action potential within the theta cycle is not affected whatsoever. So this is specific for perisomatic. I cannot say anything about axoaxonic or axonal segment inhibition, but at least in these two comparisons, timing is assured by this particular class of neurons, which are parvalbumin basket cells. To give you a better explanation or easier explanation, this is just one single session. These are the preferred phases of the place cells. This is the theta cycle shown twice, so this is not distance or time, this is theta time, or actually theta phase. And you can see that every single slot in the theta cycle is occupied very effectively and nicely by a neuron, and each of these neurons have partners that form cell assemblies. So the cell assemblies are put into slots of the hippocampal theta phase. After removing the perisomatic inhibition, this is all gone, or most of it is gone, and the neurons tend to fire around the trough. So if this is very important, and this is what I try to show you, that how important it is to have this tau-T relationship, how important it is to keep this relationship intact. If you break it down, then instead of allowing the neurons and assemblies to carry information, this is what is left. Now, this is all physiology. Very nice local manipulation. Does it have any consequence on behavior whatsoever? This is what I'd like to know. But our perturbations were very local, and that was explicitly done, so we didn't want to interfere with the rest of the hippocampal circuit. But of course, this doesn't have any impact on behavior. So now we have to ask, if timing is so important, what happens if we scramble the action potentials by some means? And we are working on it. This can be done with optogenetical methods, very nicely analytically. But for now, we don't have that, so we go back to the rat and use something that I usually don't like to use, but effective. So the nice thing about marijuana for a physiologist is that the firing rate of the pyramidal cells and interneurons don't change if you use CB1 receptor agonist in the recreational dose of marijuana. In other words, if you put the animal or a human into an fMRI, you probably wouldn't see very much change, at least not because of the firing rate changes, because they don't change in a second time window. But they do change within the theta cycle. So then the question happens is, what happens to the place fields? Right? And the reason why we ask this question is because we know that after marijuana there are memory problems, and other people before us have shown repeatedly that the hippocampal, that the animal under the influence of marijuana has problems in various memory tasks, including spatial learning. So now we look at the place cells and said, oh, this is controlled, and what you see is that there's no change. And we have about 85 others, and they are all identical. So that's an interesting situation. We have a hippocampus with a complete, perfect, good map. But the same animal, which has a good map, cannot navigate in space. And one potential interpretation is that they don't communicate with each other very well, or the downstream observers that interpret the hippocampal output cannot interpret this because they interpret their output in chunks of time, typically in a hippocampal theta cycle or in a single gamma cycle within the theta cycle. And that to demonstrate that there is some trouble here, if you look at the timing of this single cell relative to the ongoing theta cycle, then you can see that, oh, the phase precession, that is the exquisite relationship between where the animal is and which phase it spikes, or which time it spikes relative to another neuron, is completely lost. And this is the group data. If you look at it more carefully, then and summarize it, then you will this allows you to make the statement: the more you desynchronize, the more you take away. Even desynchronization is not a good term, but the more effectively you break down the temporal relationship within the theta cycle, the more it will interfere with the animal's ability to remember which arm it came from and which direction it has to go. So to summarize what I try to convey today is that indeed we have a dedicated structure, a single module in the brain, which is our search engine. It's a personalized search engine, much more effective than Google, that allows us to navigate in the information space of the neocortex. Now, the way it was developed probably is initially it was designed to measure distances in the physical world and allow the animal to make shortcuts, bypasses, and detours and go from anywhere to anywhere else, without any change in the physical and physiological properties of the system. The same algorithm can be used, this is my hypothesis or claim, for navigating in cognitive space, that is traveling back in time and traveling forward into the future. And the reason why it is so good is because we have this large, very effective CA3 region. I haven't talked about CA1, I haven't talked about the dentate gyrus and all the other complications, but this is what is, for me, probably the most prominent feature of the hippocampus, this large random connectivity matrix. And it has a proper coding mechanism, a temporal metric, which is about the theta cycle. And this is it. But of course, I didn't do it all by myself. There are many, many people who are involved here. The most critical person whose name is not here is Sebastian Royer, who did all the optogenetics, which was a collaboration with Jeff McGee's lab. Thank you very much. Go ahead.
So I have two questions. One is very specific and the other is more general. The specific one, I was wondering if you could go back to a slide where you're talking about the PV parasomatic inhibition decreasing over the place field and the dendritic inhibition increasing. Yes. So this is inhibition. Do you know that the parasomatic inhibition that's changing over the course of the place field is actually from the PV neurons? The reason this is confusing to me is because if you record from PV neurons, generally you expect to see sort of this 50 hertz firing rate from them. There are other cells, basket cells obviously, that project to near the soma, the CCK cells, which seem to be would be more likely to be changing over the course of, say, a theta cycle or at the time course of a place field. What are your thoughts on that?
So the comment is that if you look at a basket cell, a parabolic basket, it typically fires homogeneously. You don't see a lot of relationship between where the animal is and how the cell fires. And then the second question, which is embedded into this and more important physiologically, is that where does this change happen? If it does, it's between the place cell and the basket cell, or the basket cell and the place cell. The answer, I don't know. But what I know from physiology is that pyramidal basket cell synapses are depressing. This is the case in the neocortex, as Henry Markram has shown it repeatedly. Nice woman in London. Several laboratories who have shown this, that indeed this is depressing. Scanziani at UCSD has even went further and looked at perisomatic and dendritic targeting inhibition. And he sees something like this. In fact, over the course of 1 second, he stimulates the circuit and looks at what happens to the recruitment of the basket cells. The basket cells behave just like this. And the somatostatin cells, the dendritic inhibitory cells, are recruited. Thompson is the woman I have in my mind. So even just these 2 synapses are sufficient. On the other hand, there is good physiological evidence that the perisomatic inhibition from PV cells is also depressing. And there is also recent evidence from 3 labs that the somatostatin-containing neurons, at least a group of them, is innervating the basket cells. So there are at least 3 sources where this could be explained. Having said that, there is another way of thinking about, namely that your lab, Carol Barnes's lab with Andrew Mora showed first, and then our lab and also Matt Wilson's lab showed that basket cells every now and then also show beautiful phase precession, especially, and they could show also, and it's very clear, that it is because they are enslaved to a pyramidal cell that is a place cell. So even though a basket cell collects information from various places, if you are in a small volume of hippocampal tissue, then there are very few place cells. So one can dominate, and that can enslave that interneuron, which will oscillate at the frequency of the place cell, which is faster than the ongoing cycle, which means that the soma of the assembly that it innervates also gets inhibition at frequency faster than the ongoing cycle.
But part of that question is, do we know that those are really the PV basket cells, or could they actually be other neurons?
Here?
Well, with the phase precession and
I know for sure because I silence them. Yeah. And I look at their activity, and we know black and white from our experiments after the Mora experiment is that I was dealing with the palpable entitled basket cell because I looked at the phase of theta, I looked at how it behaved during sharp phase, and Peter Shomogy has identified them. So by 90% accuracy, I knew these were the basket cells of the PV type. And I have no knowledge about the CCK cells.
So the second, sorry to keep going, but the second more general question I had has to do with the place fields firing in the hippocampus fire differently often when the rat has to make a right turn or make a left turn. But there are plenty of cases that you can observe that the rat has perfectly good memory or without seeing a change in the code of the hippocampus on either in a waiting period or on the rat's way to the choice point. And likewise, there are also studies that are more recent. I don't know that they've been published yet. That can show that the place fields can really be very different, and yet the rat's memory of what space it's in and what's going to happen in those spaces is perfectly intact. And so part of the question is, the first part of this, there's a solution to, which is even though the place fields may be the same on the way to a right turn versus a left turn, there may be very brief, such as what Reddish's lab has shown, brief periods where the hippocampus, perhaps on a ripple or something, is actually, the cells are firing, indicating to the rat, reminding the rat, as you may say, where it's been or where it should go to. Even, that might be separate from quote-unquote place fields firing. And so the general question that all this comes to is, do you think that there's something to be said for what the place fields are indicating in terms of, well, I want to say present tense, but what I really mean is very recent past and very, very soon future and present tense, because you have the past and future slide there. But in the general vicinity of the present, and that the sort of more the farther back past or the farther forward future may actually occur outside of quote unquote place fields might occur, say, outside of theta, actually, is what I want to say, on ripples that you wouldn't call necessarily synonymous with theta.
So there are 15 questions here.
I'm asking
the first, I understood it. So I have to, it's a good excuse to remind myself, is that these direction predicting cells were first discovered by Eichenbaum's lab, and then Matt Wilson, I don't know which one was first, but about the same time. So indeed, there have been, there has been an underground movement for a long, long time that indeed it's not only anchoring neurons to the environment that is going on, but something else, and it has something to do with memory and so on. And the place cells are very plastic. Michael Hasselmo had a nice experiment showing that they are shifting from the beginning of the session to the end of the session in a predictable way. This experiment with the CB1 receptor shows that you have got a beautiful place map and terrible behavior. Catherine, somebody, University College London, she showed very nicely that if the animal has to learn a memory task, but you change the environmental cues, then the place fields look different. The entire map is different, yet the memory is perfectly right. This is the point you wanted to make. The truth is that all these things can go on a multiple level, multiple times, and it comes back over and over again, this same question is, what does the hippocampus do, and why this complicated thing here when this task is doesn't even require the hippocampus? Now, let me remind you that phase precession by O'Keefe was discovered in a track where the animal runs back and forth. It doesn't need the hippocampus. The hippocampus is active all the time. It just can't help. For me, it's like a sentinel. It's like a parking lot camera that is watching the parking lot for 24 hours, 7 days a week, and then it's all wasted, except one time when your car is stolen. So I think these dynamics are useful because it means that there is a constant communication of the hippocampus between the neocortex and the hippocampus itself, even at times when there is not useful information being sent out. I have a little problem with the idea that, oh yes, we have shown it also that with a ripple you can predict what the future is. We could predict what the future is by looking at the first second. But if there would be nothing else down the road, there must be a mechanism that connects this information all the way to the animal's legs, which turns the animal into one or the other direction. So I wish I could tell you how it exactly happens, but in fact, it probably doesn't matter how this trajectory is made together, and there will be multiple representations. How many trajectories can we form, how similar they are, how they interact with each other, this is all something unknown but addressable.
Yeah, I'm sorry for not being more simple and asking the question. Part of what I'm asking is, do you believe that there's something, a different state that the hippocampus is expressing when the hippocampus is encoding the state of either environment or the state that it's in versus when it needs to retrieve something?
The answer is definitely no. There is no, this is Michael had an interesting model. This is, we learn and then we recall. We learn in the class and then we recall during the exam. This is a nice temporal separation. The answer is no. There is no learning without recall. This happens simultaneously, and in my mind, it always happens during the theta state.
Thanks.
Yes?
So from the perspective of memory in general and encoding ongoing experiences, the kind of data you beautifully show seems to raise some kind of puzzle that we're somehow parcellating up our experience into these little data-sized packets that from one cycle to the next could potentially be different or overlapping or something else. So I'm curious how, if you divide your experience up in this kind of way, doesn't that raise some challenges for encoding memories across longer timescales, for stitching back together whatever you just divided up? So I'm interested in your thoughts.
So this is a very interesting question. How can a quantized information look smooth? And the answer is, to me, simple. it depends on the observer. If the observer has no way of telling or resolving time within this time frame, it doesn't really matter. For example, in a gamma cycle, this is my personal opinion, it doesn't really matter what the sequences of the neurons are. In the first approach, I can give you exceptions about it, because the membrane constant is the integrator. Now, in the theta cycle, it does make a difference. And the theta cycle is, in fact, quantal. It's not a smooth transition. We just had a paper recently where we showed that how much we can improve the relationship between the animal's position and the theta phase if we take into consideration the true phase rather than approximating it with the sinus. So the other thing also is true if you take the animal and said, let's predict the time, how long time the animal has been running in the wheel from the evolving sequences. If you look through time, or Weber time, or anything, that is not so effective. If you take cycles, then it turns out that the time doesn't degenerate. It's because in every single theta cycle, you recreate maximally that what you can, and that allows you to recreate with the help of the CA3 system and make it more complete. Now, why don't we feel the quantal resolution? One thing is that it could be that indeed the hippocampal theta cycle works just like we have a saccadic eye movement, and then your terrain is moving, and then the entire world is moving, and then you have a saccade and you've got a blank. You don't feel it because there is a mechanism in my brain that suppresses it. So this is one argument. The second argument, everything I said so far today was about the dorsal hippocampus. If you move to the posterior ventral hippocampus, the place will get larger. This quantalization is probably different a little bit, I don't know. And if the outside observers observe different segments of the hippocampus together, then they can be smoothed. I don't think that's the answer, by the way, but that's a possibility. Even longer answer. Recently, I heard a very nice story from the Moser group. Grid cells, I don't know if you mind, that they looked at the size of the grid cell. Said, what is the size of the grid cell? The reason why I'm telling you is exactly the same question came up to us in our group. I said, well, there is a small grid, bigger grid, and even better, it's a continuous change, or is it quantal? And the answer is, there are only 4 quanta. And this is sort of a good news for us, if it's true, because we found that in a theta cycle, there are at least 2 different gammas. And there is slow gamma and faster gamma. There are about 4 gamma cycles, 4 slow gamma cycles in a theta cycle, which means that the phase precession, place field size, the grid size, equals phase precession slope. Phase precession slope equals the phase of the neurons, but the phase of the spike, the phase of the spikes cannot be smooth because the theta is not smooth. There are these gamma bumps. And if there are 4 gamma bumps, then there could be 4 bumps. So maybe the idea that, yes, there is a quantalized resolution. This is inevitable because oscillations are about chunking times and silencing neurons. So it's a quantalized way how everywhere where information is coded, how it is translated to experience, Ian Gold will tell us. So the short answer is, if you read the paper, the answer is there. But so the trick is that how many quantas you can put together is determined by the relationship of this theta gamma, which is the tau versus t relationship. But it can be changed differently, as you are pointing out, namely that we can increase the inhibition, if you want. We can increase the delay between assembly 1 and assembly 2, the result of which, if it happens over the board for all neurons, then the theta cycle can go up to 4 hertz. Right? If it's 4 hertz and nothing happens to the gamma cycling, then there are more gamma cycles in a longer, slower oscillation. And this could be a reasonable question. For example, recently we showed in the prefrontal cortex there is a 4 Hz oscillator, probably coming from VTA, which is interacting with the theta cycle in a 1-to-2 interlocked manner. So they work together, but to your point, there are more gamma cycles, the more slots are available in the prefrontal cortex than in the hippocampus for holding information probably for a longer period of time. So yes, there is a way how we can prove that this is relevant to everything else. One way would be to trick the system, slow down the dynamic of theta, for example, that all of a sudden from the medial septum, for example, everybody would receive a slower timing, such as Michael Fee has shown in the songbird, for example. And we don't have the answer to that. Or doing it locally. What would happen if you just, everybody else is doing the same thing, but then you change it locally. Incidentally, you can do it another way, and I think it was the reflection to the previous question is that the future is not necessarily predicted, the presence, by the size of the place field, because there may be a subthreshold membrane oscillation prior to the first spike that is already part of the place field, or even after, when the spike is no longer there, there could be something. So what you see is the tip of the iceberg. We could reject it, I think, reasonably well from our Parvobon experiments, because this is exactly what we were looking for, that when you take away inhibition and the neuron fires more, what does it mean? That there will be spikes where they were not? And the answer is no, because the place field size did not change whatsoever. So either that is a very smooth time, or indeed it is the timing between one assembly and the other assembly that exquisitely determines the lifetime of the neurons. It's a little confusing what I said, I realized.
Okay, so I'll just remind you quickly that there are snacks and coffee and so on up in the room over here, and we'll reconvene in about 15 minutes to finish our hippocampal morning in brain day. But if you'll join me once more in thanking Jude.